Okay. Now it's my great pleasure to introduce our distinguished keynote speaker, Dr. Xihong Lin. Dr. Lin is a professor and a former chair of biostatistics and coordinating director of the program in quantitative genomics at Harvard Chan School of Public Health, as well as professor of statistics at Harvard University. She earned her PhD in biostat from the University of Washington and has made a seminal contribution to biostatistics, statistical genetics, and public health. Dr. Lin was elected to both the National Academy of Medicine and also National Academy of Sciences. She has received numerous prestigious honors, including the Katz President's Award, the F.N. David Award, and the Jerome Sachs Award for Outstanding Cross-Disciplinary Research. So please join me to welcome Dr. Xihong Lin.
Thank you very much for the kind invitation. This is my first time attending this conference. I feel very honored. I hope I'm not the only statistician in this room, and so I'm ensure there are not many formulas. And so I think it's really important to build a bridge between statistician and the computer scientist. I feel really appreciated that you all make me feel welcome. So I'll share with you some of the thoughts and I have had in the last couple of years and how to bridge the statistic and the generative AI and the genomics health to accelerate the discovery. So let's start from ChatGPT. Everybody knows ChatGPT is that really a tipping point of AI. And nowadays, we deal with much more complex data. Besides the traditional structured data like rectangle data matrices, and there are many types of unstructured data such as text, images, and also voices. And then the data become much more complex and also much more heterogeneous. And also there are many emerging machine learning and AI method which has emerged in the last couple of years. For example, large language model, transformer, and also the diffusion model. And so with all those exciting opportunity, an important thing in scientific discovery is how can we make scientific discovery trustworthy? And so give you example. So if one asks the question in ChatGPT, say how many pathogenic variants does BRCA1 have? And then we all know ChatGPT has this hallucination problem. So ChatGPT will answer about 1,000 to 1,200. But if one uses the ClinVar database, they are only 92. And so that means we all know that ChatGPT sometimes make up answers. And so therefore, trustworthy is critically important. And another limitation we face in academia is compared to industry, and the computational resources we have in academia is much less, and in terms of number of GPUs. And for example, many universities have 100, if luckily 1,000, but like Meta has like almost 350,000 GPU. So how can academia find a sweet spot and in this dimension? So how can we set up a goal? And so my view is the goal is to how can we make an impact in science by solving big problem. If this is the goal, then it's more likely for people with a different discipline to collaborate and to jointly solve the problem, because different disciplines have different expertise. And so each group, how can each group, for example, for me as a statistician, bring to the table and to an AI table to accelerate scientific discovery, how the computer scientists bring to the AI table to accelerate real world discovery. And so in order to do this, it really needs a cross-discipline expertise and for people to work together. In particular, this conference is focused on biology and health science. We all know in biology and health science, because it's, the biological systems are complex and also affect life. So therefore, the scientific discovery and also analysis result have a high bar, and so for discovery, and because it affect people's life. So therefore, the field generally are more conservative, and because it affect people's life, and also in order to make the translational science, and so the discovery has to be trustworthy in order to push to the translational space. So how can we do that? How can we celebrate the true discovery? And so I view there are two-way street. And the first is how can we empower statistics and also the analytic application in the real world problem by leveraging AI transformative potential? Also, how can we empower trustworthy AI by leveraging rigorous statistical principle and also accounting for the randomness and uncertainty? So this really calls for the joint effort between the statistical community and the computer science community and also domain science community. And so, for example, if one look at the ChatGPT answer, the ChatGPT answer can be wrong. So how can one create a confidence score for the ChatGPT answer to address this hallucination problem? So I'm going to focus on the first part. How can we accelerate the discovery by leveraging statistics and generative AI and domain science? So in order to do that, it would be useful to think about how we can build a data science ecosystem. And so in my view, it has three pillars. One is the diverse and the fair data, and also statistic, machine learning, and AI method tool and infrastructure, and also how can we make the results interpretable. So in order to achieve this, is useful. I tell my students to become a full scientist. So that means not only strong in quantitative science, like statistics, computer science, but also strong in domain science. So this will help with the ability to identify big problem and also improve communication skill. So in communication between domain scientists, statistics, and computer scientists. Historically, a statistician work with the biologists and the clinician and the public health researchers, but historically, and statistician and computer scientist collaboration is less, but nowadays it's become more and more critical. And so it will be great to build this three-way collaboration. So with this shared common goal is how can one solve a big problem and in science and by working together. So this work, this presentation has two parts. The first part, I'm going to talk about how can we empower statistical genomic and scientific discovery by leveraging generative AI and statistics and using the synthetic data. Second part, we'll talk about how to build an ecosystem and at scale. So we all know, like machine learning and AI, the primary focus is prediction. However, prediction is not everything. And the influence on population parameters, for example, effect of genetic variants on the human diseases, and so that can help identify the drug target. And this is equally important. And to understand the science, understand how the biology works, how to use a genomic, identify the potential drug target, how to develop intervention strategies, and this will help taking action. Because if one use the genetic profile, say predict a person's disease risk, and then a patient will tell the clinician, I cannot change my genetic profile. My mom and dad give that to me. Then what can I do? So therefore, one needs to think about action. So therefore, the inference is critically important besides the prediction. And also, how can we empower statistical analysis and also scientific discovery by leveraging generative AI, such as transformer and diffusion model. And so in order to do that, so one thing we need to keep in mind is those generative models can be wrong. So how can we still have the true discovery, even though the generative model can be wrong? But if generative model is useful, that can empower statistical analysis and empower scientific discovery. So in order to do that, this generative AI-generated synthetic data can offer help. So this is a very nice talk. Many of you heard this talk, and by Leah Satkava last year in Europe, 2024. He made a really interesting comments about the pre-trained data and the synthetic data. You know, he is a former chief scientist of OpenAI. So he talked about open wall, so that pre-training will end because there's only one internet, and the data are not growing. So he conjectured 3 future AI development. The first is the synthetic data, and the second is AI agent, and third one is the inference time computation. So I was very glad that he mentioned the synthetic data. So how can we leverage synthetic data? So first question we want to answer is how to generate the synthetic data. And so there are multiple ways. A traditional way is using the parametric model, for example, using a linear regression or logistic regression, or use a non-parametric model, for example, like random forest, XGBoost. And in recent years, there are multiple other ways to generate the synthetic data. One is many of you are familiar with is variation autoencoder. And a key assumption of variation autoencoder is it need to have a low dimensional representation. For example, you start from this image and then run the encoder and in some latent space, and then that does the dimension reduction to have a low dimensional representation of the data and run the decoder and to regenerate the data. However, not all the data have low dimensional representation. And so in the last couple of years, and then also transformer has been a popular tool. And so many of you know, like large language models such as ChatGPT basically is built upon transformer. So for example, if one input English sentence, how are you? Running the transformer will translate to a different language. So that can help to generate the new and contextual coherent samples. And then the assumption there is the data have to have some kind of meaningful order or the sequence. And so for example, in a sentence or a DNA sequence, and then the transformer will be useful. So many different transformer-based foundation model have been built in the last couple years. For example, like an informer that is based on the DNA sequence model, or any kind of X-former, and really nicely provided by Meta. One can basically customize it using any of application you are interested in. Another one which I'm quite passionate about is the diffusion model. So diffusion model has 2 processes. One is the forward process, and so this process doesn't involve any training. So basically, you start from an image, and then you gradually add noise until it becomes a complete noise. And so this part has no training at all. So the art is the reverse process. That is the generative process. So basically, you start from the white noise, and then you gradually denoise it using the neural network or the deep learning until you recover the image. And so this part is art. Then how can you regenerate the data? And there are many different conditional diffusion models. For example, you can do change the background. That basically is an example of the conditional diffusion model. So the benefit of diffusion model is it can learn the complex distribution of this high dimensional data, and that may not be ordered or may not have low dimensional representation. So this is quite attractive. And there are drawbacks. And for example, the sampling are computationally more expensive. And so this is really an active area of research, for example, using the UNet. So to illustrate, and so the diffusion model basically tries to learn the distribution of high dimensional data on X, and that generate the observed data or transfer the high dimensional data. For example, change the background of that the image of that cat. So for example, suppose the original data is this bimodal normal distribution, and the forward process, basically, you gradually add noise to become a normal distribution. And then the reverse process is you do the deep learnings and then regenerate original data. This will recover the original bimodal distribution. This is just one-dimensional example. Then how to do this, and how to do denoise, and that basically is the art. So you start from the, So you start from the white noise, and then basically what you do is you try to predict the noise, and then each time after you predict the noise, you subtract noise, and then predict the noise again, subtract noise, and go on, and then until you recover the image. So how can you denoise it? So basically that is where the deep learning comes in. So basically in order to calculate the predicted noise, and you train using the white noise was the standard function using the score matching and using either the data at the current stage xt, and then you try to predict the white noise or predict the score and using the deep learning. And so that is where the art is, and also the computational challenge lies in here as well. Then a natural question we ask ourselves is how can synthetic data from generative AI be used to empower statistical analysis and ensure trustworthy scientific discovery, even the generative data can be wrong, and the generative model can be wrong. And so I'm going to use this paper as an example to illustrate the idea. And so this work was led by my former student, Zach McCall, and also Jessica is our former alum. And this paper is Zach's, one of the Zach's dissertation paper, and was published in Nature Genetics 2024. So I'm going to use the UK Biobank as example. So in UK Biobank, so because we have a genetic data, we have an electronic medical record, we want to see how the genetic variants associated with the human diseases. However, many of the phenotypes, and for example, body composition, very expensive to measure, is only available for less than 5% of the population in the UK Biobank. Then the question is, how can we leverage AI generative model to generate the synthetic data to empower the analysis? So to illustrate, suppose the target phenotype is a body composition that is measured about 30,000 people. In UK Biobank, total sample size is about 500,000 people. So you can see over 90% of the body composition is missing. That is marked by Y. And this S basically is a demographic variable, biomarker, and so on. That is available for all the UK Biobank sample. If we restrict to independent European sample, this is about 350,000 people. And then also we have the genotype data and from the GWAS, the genotyping, and that have over 1 million variants. So what is the goal? The goal is to study the association between the genetic SNP and the phenotype Y body composition. So you can see that if we only using the 30,000 people to do the analysis, it's not much power. And but we have a lot of good predictor to predict body composition. Then how can we leverage those surrogate information to improve the analysis power in studying the association between genetic variants and the body composition? So the goal is, let's predict body composition using the surrogate. So how can we do that? There are multiple ways. One is, suppose you have a pre-trained model and with a external pre-trained weight. And for example, using the transformer or diffusion model that generate a pre-trained weight. And then one can fine-tune it. And this nowadays is what most academia does, because train the model is expensive, but if one fine-tune the model using existing weight, it's much cheaper. And then you fine-tune the weight and then generate the weight suitable for the UK Biobank sample. And or you can split the data and then you create a part of the labeled data, for example, 5,000 people with observed body composition to train the model to predict body composition. And so here, then suppose I train the model, use 5,000 sample, get the weight, and I apply that weight and to the whole 500,000 people in UK biobank sample, then I predicted body composition for everybody. Then I have Y hat for everybody. So this shows a picture. Suppose like orange represent observed body composition, only 30,000 people, less than 10% of people with observed body composition. But I have many covariates like age, gender, biomarker, and also EHRs, and that can be used to predict body composition. That I can predict for everybody. And then now I'm going to leverage the Y and the Y hat to study the association with the genotype. And so wrong thing to do is after you calculate the Y hat, you study the association between X and the Y hat. That will give you biased result. And because the predicted body composition can be garbage if the generated model can be wrong. So therefore, the natural question is, how can we make a valid statistical inference if the black box AI generated model is wrong? My hat is completely garbage, and I still want to ensure my association between the genetic variance and the body composition is still correct. But if my generated model is correct, and then I will have a better power to study the association. Okay, so what are the traditional analysis strategy? The first is just use the label data. So basically I have an orange that is 30,000 people, and then I look at the association between Y and X. Or I can do the simple or multiple imputation. So I use the orange is 30,000 people with observed body composition. For those people don't have body composition, I impute them and by the Y hat. And then you can do this once, or you can do it multiple times. That is called multiple imputation. And now you use the stack data to run the GWAS analysis, look at the association with the X. And so you can see in order for this to do well, the generated model has to be good. If the generated model is wrong, this multiple imputation is likely to be wrong. A third approach is what proposed in this Nature Genetics paper by Zach. So we do the synthetic surrogate analysis approach. Basically, we do the joint analysis of Y and Y hat on X and accounting for the fact that the generated model for Y hat can be wrong. So how can we do this? And suppose I have 5,000 people I use for train the model, and then I have leftover 25,000 labeled data, and also the rest of data is unlabeled. And so those are my data. We do the joint modeling. So you use the Y and Y hat, and so you have a GWAS model regress on the genotype, and then you have a Y hat also do an association with X as well, and you model the correlation between them. So this basically, if one look at this correlation here, this basically measure how good the predictive model is. If the predictive model really good, then this correlation should be close to 1. Okay, but in reality, it can be garbage. But we want to do the right GWAS analysis association between Y and X, accounting for the fact that generative model can be wrong. And so, but turns out, instead of running this data analysis, joint analysis, accounting for missing data, and you can do this simply using the sequential univariate analysis without additional software. So that makes analysis much easier. So this can be easily implemented in the following way. So first, what you do is you run the GWAS, regress the Y hat on X. And so then we know this can be wrong because Y hat can be garbage. That's okay. And then after I do that, I calculate the E hat, calculate the residue. And I do that for all the data, all the 500,000 people. The second step, this is where the calibration comes in. So I do the conditional analysis and using only the labeled data. So you can see that here I regress the Y on the residue calculated from the first step and also on the genotype. And then by doing that, you can see I calibrated. Even the Y hat is garbage. It's perfectly okay. It doesn't hurt me. And I always have the positive learning. And then my coefficient of the X will be the right coefficient of the association between Y and X. And also then this result is identical to that joint analysis, and also it has the following properties. And so I'll show the property in a minute. So basically, this is a workflow. So first, you has 5,000 people for training, and then analysis, I use the rest of part of the data for analysis. For the training, I basically use the covariate to predict the body composition, use the 5,000 people, any kind of model you like, and like a random forest, XGBoost, diffusion model, whatever model you want to use. And then you predict the Y. And then that gives you the weight. Then you transfer the weight to the whole UK biobank sample, 500,000 people, and you calculate the predicted body composition for everybody. And now you have this labeled sample and also predicted. This is your synthetic data, generated synthetic data for body composition for everybody. And then you run the GWAS analysis, analyze each SNP at a time. You got this Manhattan plot. And the one here will show the potential association with the SNP. So you can see this is very easy to implement. What are the properties? And the first property is it is robust to misspecification of the generative AI model. And so if this model is wrong, I'm okay. And also this generate a valid statistical association between the SNP genotype and the phenotype. And so under the missing at random assumption. What does that mean is basically the labeled data and unlabeled data, I don't need them to have the same distribution. And so this can be easily implemented using this sequential GWAS procedure I showed you. To show you here, that is the, so here, this is the generative AI model. Suppose the generative AI model for Y is correctly specified, and then you can see the standard approach using complete data and also the imputation approach and also synthetic surrogate approach, they all give unbiased result. However, if the generative model is incorrectly specified, if you do the statistical procedure right, and then you can see the synthetic surrogate approach give unbiased result. But if one uses the imputation method, you can see you are going to have a biased result. So the second property is always positive learning. So what that means is this synthetic surrogate approach is guaranteed to be more statistically efficient or more powerful than using just the complete data analysis with the 30,000 people. And so the power gain increase with the proportion of missingness and also correlation between Y and Y hat. So that indicates how good the generative model is. So that is good. So there's no negative learning. So that means even the Y hat is a garbage, the result will be similar to what you use using just observed data. But if Y hat is useful and you are going to have a smaller standard error, you are going to have a more discovery. To show you the finding, and so here is analysis and using different body composition. So for example, if we use the leg as example, if I only use 30,000 people to do the discovery, you can see this is the number of the SNP which have been found to be associated with the body composition. But if I use this synthetic surrogate approach, you can see there are much more discovery. A natural question is, how do we know they are the true discovery? More discovery doesn't mean may not always be good. It can be false discovery. So therefore, benchmarking is critically important. So we compare the number of the discovery with those reported discoveries in the GWAS catalog to see how many overlap we have. So the results are encouraging. So you can see this is the number of overlap using the standard approach, just using observed 30,000 people. And this is the number of discovery and overlap with the GWAS catalog. And as you can see, there's much more overlap. This orange is the difference between the blue and the red. So this is a new discovery, which are replicated in the GWAS catalog. So that is good. So you may wonder how much efficiency gain you will have. This can be calculated using this formula. Is called asymptotic relative efficiency. Basically, you compare the variance of the two estimators using the standard approach, using the 30,000 people, and also using this synthetic surrogate approach. Its formula is very simple. It is a function of the proportion of a missingness and also the rho that is a correlation between Y and Y hat that indicate how good the generative model is. To illustrate using example, suppose 99% data are missing, only 1% data are available. Suppose my generative model is perfect and rho is 1, and then you can see this relative efficiency is 100. What that means is variance reduction is 100 times. So that means you effectively increase the sample size by 100 times. However, if the rho is 0, that means my generative model is wrong, it's a garbage, then you can see relative efficiency is 1. That means the result will be the same or similar to those using only observed data. So there's no negative learning. And so here is an example. So you can see if this y hat is useless, the generative model is a garbage, and then you can see there's no bias, and the relative efficiency is 1. So that means the standard error is the same, just use the same complete data. However, if the generative model is useful, suppose the rho that's 0.75, that means R squared is 0.5, then you can see that 75% of the data are missing, and then you can see the relative efficiency 2. That means you increase the double, effectively increase the sample size, doubled it. So that is good. And then some of you may have heard about this PPI, and this is a very nice paper published by Michael Jordan's group and he and his students. This was in Science, published in 2023. I did not know this work until I gave a talk last year in the fall at Stanford, and Rob Tishrani mentioned the synthetic approach we presented is like related to this PPI work. And then he sent me the paper on the way back from San Francisco to Boston. I took a look at the paper. It seemed like they are indeed related. I'm going to show you the differences. And there's another paper which was published later on after our paper and called PopGWAS. And then I'll show you how they are related either. Too. And so this work was led by my postdoc, Ruoyu Wang. So let's start from a PPI. And so in the PPI has this very simple formula. Suppose you have the regression. You want to study y association between y and x. And then suppose you have y hat. That basically is you just regress y on x using the labeled data. That is your beta hat. And then in order to calculate the PPI estimator, so what you do is you use the y hat and on x using the labeled data that was alpha hat labeled. And then you use regress y hat on x using unlabeled data. This was alpha hat unlabeled. You take a difference of this and then you subtract from this beta standard. And so you can see the formula is very simple. By looking at this formula, you can immediately see the problem. First, the generative model has to be good, right? If generative model is not good, then all the alpha will be biased. Second is the labeled data and unlabeled data have to have same distribution, right? If they don't have the same distribution, and even though the generative model is correct, then you can see this difference will be biased as well. Okay, so that basically assumes two assumptions. The generative model had to be right, and also the data had to be missing at random. That labeled data, unlabeled data need to have the same distribution. And to fix the first issue, to allow the generative model to be incorrect, and this is what PopGWAS does, and then basically multiply this difference by a little constant that model the correlation between y and y hat, model how good the generative model is. So what are they in common? And all the three methods are robust to the generative model misspecification and the missing completely random. That means labeled data and unlabeled data need to have the same distribution. So what are the differences? And both PPI and PopGWAS are not statistically efficient, and they have a larger standard error and were lower power. So however, the synthetic surrogate approach is statistically efficient at the smallest standard error, so basically the highest power. And we call something called the AIPW estimator. And then also the PPI can have negative learning. What that means is that if the generative model is wrong, the standard error from using the PPI can be bigger than the standard error just using the observed data analysis. And so if the generative model is wrong, so that is not desirable. However, if one uses synthetic surrogate approach, it's positive learning. So that means the standard error of using the synthetic surrogate approach from the GWAS analysis is always smaller than using the complete data analysis. Even the generative model is misspecified. So that is good. And also, both PPI and PUB/GWAS require missing completely at random. So that means the labeled data and unlabeled data need to have the same distribution. However, synthetic surrogate approach only require missing at random. That means we don't need the labeled data and unlabeled data to have the same distribution. So that is useful. And to show you, suppose the data are missing completely at random. That means the labeled data and unlabeled data have the same distribution. And so here, this is a standard approach, and this is a PPI, and this is PUB/GWAS, and this synthetic approach. As you can see, all the methods have unbiased estimator. However, you can see the synthetic approach have the smallest standard error compared to the other approaches. Furthermore, if, so you can see the PPI have a bigger standard error. And furthermore, if you compare the PPI with the standard approach using completed data, you can see it has a bigger standard error. Because here, the correlation of the y and y hat is it's about 0.8. That's already pretty good, but you still can have a negative learning here. Now, if you have a missing at random, that means the labeled data and unlabeled data have a different distributions, and then we are going to have a problem with both PPI standard approach and also PUB/GWAS approach. You can see they are all biased. However, the synthetic surrogate approach still unbiased. So that allows the labeled data and unlabeled data to have a different distribution. To summarize this part, so the machine learning and AI generative synthetic data are powerful with many potential. I just use this GWAS analysis with the missing data as an example, but it has really this kind of synthetic approach really have lots of potentials. But you have to do it right. And so basically that is where the sweet spot is. By marry generative AI with the statistic, and that can empower statistical analysis and with the missing data, but also is allow for the generative model to be wrong. And so this offers a more robust and powerful analysis than both the PPI and PUB/GWAS, and so it ensures positive learning. There's no negative learning here. So let's move on to the second part of how can we build the end-to-end analysis ecosystem. So I'm going to use the whole genome sequencing study as an example and to help build this ecosystem. So this will involve the whole genome various functional annotation and also build a whole automated analysis pipeline and also deploy it in the cloud. The reason is many of those large whole genome sequencing data and biobank data are only available in the cloud. And the cloud sounds wonderful, but they are expensive. We need to make it affordable. So this will involve a data science engine and also engineering as well. So this is basically a quick overview of the timeline of a whole genome sequencing effort. About less than 20 years ago, and that launch of 1,000 genome project, about a couple thousand people were sequenced, and because at that time the sequencing cost was pretty high. In the last couple of years, the sequencing costs have dropped dramatically. Several large whole genome sequencing programs have launched. For example, like a TopMed is by NHLBI, we have sequenced about over 200,000 people. And there's a genome sequencing program by NIGRI. We have sequenced about 140,000 whole genome, over 200,000 whole exome. And then the multiple large biobanks have emerged. For example, UK Biobank has 500,000 people with GWAS data, whole genome sequencing data, and also electronic medical record with thousands of phenotypes. And the Million Veteran Program enroll 1 million people. ALPHAS is basically US version of UK Biobank, and enrolled 1 million people, has sequenced about 400,000 people. So totally we have over millions of people with the whole genome sequencing data in hand right now. And so compared to traditional whole genome sequencing analysis or GWAS analysis, an important aspect of biobank is one bird hit multiple birds. And so in traditional GWAS or whole genome sequencing analysis, so basically one look at the whole genome, and look at a particular phenotype. But with the biobank, one can look at 1,000 phenotypes simultaneously. So that is much more cost-effective. So that's because if a particular drug target has a strong genetic basis, then the clinical trial are more likely to be successful. So that's why the UK Biobank whole genome sequencing data that are largely covered by several large biopharm companies. And so many of those large biobank and whole genome sequencing studies have a diverse multi-ancestry sample with European, African, Hispanic, and Asian. So you can see from both TopMed, GSP, and ALPHAS. And right now, ALPHAS has about 400,000 whole genome. So the data we deal with are very large. So for example, using the TopMed, we have TopMed with the Freeze 10 has about 185,000 samples, and then before QC has about 1 billion variants. After QC had 845 million variants. Majority of the variants are rare variants. For example, 45% are singleton. That means among these 200,000 people, only one person has that variance. 16% are doubleton. Only 2 people have that variance. And the common variants covered by the GWAS array only occupy less than 1% of the genome. So the goal is we want to scan the genome to identify the genetic variant set associated with the human disease and the trait that can identify a particular drug target. So then, because the data we deal with are huge, so therefore scalability is critically important. To this end, we first need to functionally annotate the whole genome. And so the genome sequencing program, and we have the functional annotation working group. We integrate the data from many different sources, ENCODE, roadmap, ANOVA, and the GTAC, and so on, create its favorite annotation database, which include allele frequency, conservation score, protein function, epigenetic score, and so on. So these have over 300 annotations, and we make this database public available. And so we annotate all possible variants across the genome. So across the genome, there are 3 billion positions, 9 billion variants. So this is a relational database, and with annotation of all possible 9 billion variants. So we use the PostgreSQL, build a database, and use a very efficient indexing to look for particular variants. Takes less than 1 second. And we make this data publicly available. So it already has over 20,000 downloads so far. And also, because not everybody is familiar with the database computational biology, and so we built this Fever GPT, which has a nice large language model interface, and using the Fever API by using the ChatGPT API, creates this large language model interface. And so we make this publicly available as well. And so you can ask any questions you like what you do in ChatGPT. For example, ask, what is the function of R7412? This is ApoE, well-known ApoE variants. Then that will use the Fever backend database to summarize the finding. And so building this knowledge-aware AGI. Or you can do all kind of a computation. And so, for example, you ask how many pathogenic variants does BRCA1 has? Because the ClinVar is part of the Fever database, it gave you 92. Remember, if we use ChatGPT, it gave you 1,000. And so you can see we cannot use a general database. We have to use high quality specialized database in order to improve the accuracy. And so we have also built a very nice interactive visualization using the Fever GPT as well. And right now, we are also building a Fever agent, so that will help the researcher to navigate the database more easily using the AI agent. And so the goal is we want to do all the analysis completely automate, and also at the scale make it affordable. And so this basically need to use the cloud. And because all the data are in the cloud right now, the traditional way for data sharing is basically data copy. So for example, in the old days, the researchers were allowed to download the UK Biobank data to our own research computing cluster. So that brings the data to researcher. And now because the whole genome sequencing data are too big, those are not allowed anymore. So the new model is to bring the researcher to the data to the cloud. For example, this basically will need the data science engineering. And so there are multiple clouds. And so the one the NIH has is called Anvil. So the genome sequencing program data and all of us data are at Anvil, which is hosted at Broad. And also the TalkMed data are about data catalysts. And so this is also a cloud platform. And all those data can only be accessed through the cloud. And in those platform cannot be downloaded. And similarly for UK Biobank, and the platform is a RAP, and the data cannot be downloaded either, and only be analyzed in RAP. So you may wonder that how big the data are. So this is show you the UK Biobank sample, about 500,000 people, and in the UK Biobank RAP platform. So if you use the VCF format, you can see the files are really, really big, has 150,000 files, and also almost 1,500 terabytes data. 1 terabyte of data saving the Google Cloud costs $300. So without doing anything, just think about using the egress to download the data to your own cluster, installing it, and it's very expensive, right? So that's why they don't want you to download the data anymore. It's too expensive. So the first thing we need to do is we need to compress the data and make it small enough for analysis. And so we create this GDS format and also annotated GDS format, annotated GDS file. So they basically put annotation, functional annotation from Fever database and also the genotype data from UK Biobank in one file. And so the compression rate is really, really good. So it's almost 1,400 times smaller than VCF file. You can see it's only 1 terabyte. And so the GDS file is for gaming purposes, but it looks like a beautiful way to store the genotype data. So this is much more affordable, basically costs a peanut to store the data. And then also we develop a series of analysis pipeline to do the analysis, and by integrating the functional annotation, weight with the whole genome sequencing data by upweighting the variance with the functional annotation. And so those are published in series paper led by these 2 talented former students. And so one is, 2 were in Nature Genetics, 1 in Nature Method, and 1 in Nature Computational Science. So basically, as I mentioned, we want to automate everything. And so make it at scale. So we have this Fever annotation database available in Anvil, Biodata Catalyst, and also RAP, 9 billion variance functional annotation database. And first one need to QC. For example, you have the UKB data, and so that's already been QC'd. For GSP data, use a Hill and to do the QC. And then also have the phenotype data from electronic medical record from UKB. And then run Fever annotator to annotate the genotype file, from example, from the UK Biobank. And then that will create annotated GDS format. It's very, very compressed. And then one can run the the star pipeline for whole genome sequencing analysis. So you can see this whole thing is completely automated. And to do the analysis, we first need to group the variants into different masks. For example, for coding variants, group them into loss functional variants, missense variants, and synonymous variants. For non-coding variants, group them into promoter and enhancer, and from encode, for example. And you can do the a sliding window approach or dynamic sliding window approach to cover the whole genome as well. When one handles the non-coding variants, because the functionality are likely to be cell and tissue specific, and so this present additional challenges. And so basically this is analysis flow. First prepare the data, you have a genotype data and the sparse GRM matrix to account for relatedness and also the phenotype. We run favor annotator to annotate the whole UK Baobang sample and across all the variants, and then create functional annotation. And then we group the variants and into different masks. For example, APOE, we group them into promoter variants, loss functional variants, missense variants, and then we run the star association analysis. And then by the end, we create an association between each of the masks and the phenotype, for example, LDL. And so you can see this whole thing is automated. You may wonder how much it costs, and we make it very affordable. And for you can see for UK Baobang 500,000 people, to annotate over 1 billion variants is only cost $6 on REP. And to fit the null model, 25 cents. And to run the whole genome analysis costs about $8. So totally it's about $10. And so we make it really affordable for the community and to do this analysis on REP. To show you, this is analysis of a top-med 66,000 sample, look at the association of the real variants and also the lipid. So all the right gene or the right mask pop up. So for example, like for loss functional variants, PCSK9, LDLR, NPC1L1, they all pop up associated with LDL. And already a drug approved targeting towards PCSK9 and NPC1L1 by FDA. And also the promoter and the LDLR promoter and the PCSK9 promoter all pop up as well. So this is all very encouraging. Remember, this is only 66,000 samples. And also to handle the non-coding variants, because an analysis annotation are likely to be cell tissue specific. So we ask ourselves, if we use the right tissue, right cell, whether we can have a better power. The answer is yes. So for example, the red indicates the number of discovery using the liver. Liver is the right tissue for LDL. And then the heart and brain are not the right tissue. You can see if you use the right tissue, and then you have more discovery. Furthermore, if we use one more relevant cell type, for example, hepatocyte, adipocyte, the number of discovery can be even more and can also have a better interpretation. So my postdoc, Eric Van Buren, developed this cell star method, and by integrating the whole genome sequencing data with a single cell functional annotation. And so this, to achieve this goal, the NHGRI launched this impact of genomic variants on function consortia. This basically is a new ENCODE. We are one of analysis center. And so it generated many, many different data at the cell level. And so this create a cell level epigenetic map by looking at identify the cell level regulatory elements and the epigenetic functional annotation and map them to gene and networks. And it generates those kind of CRISPR perturb-seq data and PRA data, and also single-cell multinome data, like ATAC-seq data, RNA-seq data from the same sample. And IGVF will create the IGVF catalog and also knowledge graph and make that available to the community. We hope to make this available to the community, the first version available, and the data make that available to the community as well next year. And this will be available at Anvil. So you can see a lot of data will be available in Anvil, and this will be all on the cloud. To wrap up, it's important for all of us to be open-minded, think out the box, and also when we're embracing the AI, and then empower statistical analysis and also discovery using the synthetic data, and by leveraging the possibly misspecified and black box generative AI model, but still ensure the analysis is trustworthy, is valid, even the generative model is wrong. And then also build this end-to-end scalable, interpretable, and also the affordable analysis ecosystem. And so this also calls for the effort to continue fusing statistical AI and also the domain science to accelerate discovery. Thank you. Oh. Thank you.
Hi, Jim Weimer, Annual University. First part of your talk, you mentioned 30,000 things compared with the big biobank. That's great. In my world that I work in, my problems, I don't have any label data. I have prevalence rates.
Mm-hmm.
Can you comment on how your techniques might be useful when I don't have direct pairings? I just have prevalence rates.
So you mean how they got the consent? Is that what you mean?
No. Like, for instance, I know that 4% of women are pregnant at any time. I don't know which women are pregnant, and I know that 10% of them are going to have hemorrhages. But I don't have any labeled data on which women actually have hemorrhages. I just have a bunch of heart rate data.
Yeah, that's right. I think many of the traditional like demographic data, they all have labeled data. It's just more like those expensive data, like for example, the imaging data, and they only have a fraction people with the imaging data, or those expensive proteomics data, only about, I think, 10% people. So those basically, those expensive, high quality biomarker data, they don't have the labeled, they don't have the label data available for everybody because it's too expensive. This is going to be true, and no matter what, right? And so most of the time, and also for multi-omic data, and for top med, for example, multi-omic data only available less than 30% of people. And so this is going to be true because they're just too expensive. It's just not affordable, right?
As a follow-up, what if I have no labels?
Oh, you take even one step further.
Yeah, right. Because you gave the first talk where I'm actually considering synthetic data now.
So if you don't have any labeled data, I think then probably if you could borrow information from other sources, and then that will involve transfer the learning, right? You can do that through the transfer the learning, and then that can also help as well. Yeah.
Hi, Professor. I have a question on the sensor. Do we assume that the the relation between y hat and the actual y has no nonlinear relationship? Do we assume that during the data synthesis, the generative model that gives us the y hat completely make the nonlinearity between this variable and the actual y vanish? Because in general, if you want to decrease the uncertainty of even if there's some nonlinearity that's present in the relation between y hat and y or any of the other synthetic variable. In information theory, if you have another conditional variable, the conditional entropy also goes up. But maybe it loses the good property that if you have such y hat, then the final model that we are going to use has no nonlinear part that is fully explainable.
Yeah, so that's a very, very good point. So that's why it's really an art to generate the synthetic data right? So this is the whole game is how can we generate the synthetic data right? If we generate the synthetic data right, as close as possible and to the true observed data, and we are going to win. You can see that if the generated model is perfect, then the efficiency gains 100 times. And if I have 99% data missing, right? So that's why this really calls for collaboration between the statistician and the computer sciences and engineers, and to see how we can develop tools and to create a better synthetic data. And so this can be using the data from the external sources and by leveraging data from many different external sources using the pre-trained weight. But also in the meantime, we have to ensure that if the generated model is wrong, and then my analysis is still right. So that's where the statistics help. So that's really a good example to show the partnership is critically important. Yeah. Yeah. So that is true. So I think like so for us, we basically, if we all, in order to ensure the analysis is valid, even like for transfer learning or do this kind of the robust analysis. And so we all need to make assumptions, right? And so there's no free lunch, but the goal is we want to make assumption as less as possible and also as real as possible that can still ensure the analysis is valid. So this is where the joint effort between statistician and computer scientists will be helpful. And also collaboration with the domain science is critically important, and to ensure that the assumption is reasonable. And in genetic study, for example. Yeah.
Thank you for a nice talk, and it's Jinping from UNC Chapel Hill. So I have a question, not a question, but more ask for suggestions, because I'm more working on sort of pervasive healthcare and mobile computing and stuff. And we also, like, for wellness and health, we also have the issue of like shortage in sort of data, right? As well as like it's hard to recruit a human subject and it takes a long duration. So we also saw the efforts from your talk, the second part especially, from like how the communities sort of efforts, right, could build up like larger database that could benefit the whole community. So do you have any suggestions or perspectives for us, like who work on more like digital pervasive healthcare, that how could we work together as an effort to, enlarge or like benefit the community? Thank you.
Oh, that's a great question, a very broad question. And my thinking is, first is that we need to, the data, I think. So, because this data fairness is critically important. And so then in old days, you can see many of the Jiva studies are basically Europeans. And though like other population sample size very small. And so then if one use, for example, the weight calculate for polygenic risk score from European and applied to the African American and Asian, the performance will not be good. So therefore, the first is how can we increase the data fairness. And so this will need a lot of collaborative effort. And so also, how can we allow for federated learning? And so because a lot of data cannot get out of healthcare system, right? And then if the data cannot get out of a healthcare system, and for each healthcare system, so how can we allow for federated learning without the data leaving the healthcare system? And so this will involve the computational community and to help through the federated learning. And also then, so nowadays, I think the other thing we found that nowadays, because there are a lot of data available from the internet, people ignore study design. And so I think as a quantitative scientist, we all know garbage in, garbage out, right? So therefore, I think it will be important for all of us to really think harder. How can we ensure study design to make sure that the study design is properly done, even with those large observational data, then that will ensure the analysis and to be trustworthy, to be unbiased. Because for both computational scientists and the statistician, if we fix the problem afterwards, after data already in, that's much harder, right? And had to make all kind of assumption. But if we can fix it upfront as a design stage, and that will help everybody. And so therefore, I think those are just a few examples. Yeah. So I think this, those are great questions. I think this we all ask ourselves every day. So my feeling is that probably how can we build a stronger collaboration between computer scientists, statistics, and domain scientists. I would think if we have this kind of a desire, and then that really will help the field a lot. Right now, I feel this collaboration between statistician and computer scientists not that enough. And so how can we make this effort and to improve that kind of collaboration? And so the learning from each other. And so I think that will really carry a long way. Yeah.
The second question is, what do you think are the most exciting future directions?
I think this kind of the synthetic data definitely is a great, exciting direction. And also probably you heard a lot about this virtual cell. Many of you heard about virtual cell. So basically design the experiment and without running the generating the data completely virtually and before really running the experiment. And so that will save a lot of cost. And so this virtual cell and basically try to simulate how the biological system work. And so this will be exciting field, but basically this related synthetic data as well. But really need to generate this kind of models and also the analysis really mimics the reality. Because biology is complex. And also if virtual cell, just one example, how can we generate a virtual healthcare system? And this is another example, right? And so this just so many exciting opportunities that really need, especially for young people, really exciting time for young people and to work as this interface of AI, statistics, and also health science. Yeah, thank you.